Okay, so today we're going to shift gears a little bit. We've been talking about various animal groups and some of the things and adaptations that they have, and run a few labs looking at metabolic rates. We're going to take a deviation from metabolism per se, but talk about some of the equipment and the historical development of the equipment that we use today to measure oxygen specifically in the environment. Now, we can measure a lot of different gases, CO2, carbon monoxide, lots of other gases that we might run into, or solvents even that we can measure really, really quickly. And we're really good at that. But what we really need to be able to do is measure oxygen when we're dealing with metabolic studies, in addition to occasionally some other metrics as well. But oxygen turns out to be one of the most difficult things to measure. And if you remember your history back in the mid-1800s or so, Joseph Black and Anton Lavoisier were working together, and they together came up with, of course, the field of thermodynamics, which is how energy is used by animals. But the real breakthrough for them was when Joseph Black discovered oxygen, right? And so we had the opportunity at that point then to measure oxygen directly, oxygen consumption. And so what we're going to do today is just kind of look at some of the ways in which we can do that. And we're going to talk about 4 specific types of approaches to measuring oxygen. So the one that has been dominant, I want to say, for probably the last 100 years is what we call the Clark electrode. And so I'm going to go up to the board here, and I can show you how the Clark electrodes work. And then I'll show you one, and we'll see that how it works. And that should not be a surprise to some of you, because many of you probably use Clark electrodes to measure oxygen in laboratories, exercises we've made in the past. So this is kind of a schematic. They're called the Clark electrode, named after Clark, who built the first one. But the other word you'll see, or the other title that you'll see associated with that, is polarographic electrodes. So the process of, or the way these things work is relatively simple. You have an anode, and the anode, if you remember from chemistry, right, anodes give off electrons. And we submerge the anode, it's usually silver or gold in some cases. We submerge the anode then into an electrolyte solution. That electrolyte solution then allows for electrons to find their way from the anode down to the second component that's important, which is the cathode. The cathode is silver or gold, and essentially what happens is, as there's voltage applied to the anode, electrons are given off, will migrate down to the cathode, which has an opposite charge, positive charge, pulls the electrons down. When they touch the cathode, they're reduced. And that creates an electron, and the more electrons that you have moving through the solution, the higher the voltage that you're going to be able to measure. So essentially what happens is we go ahead and we plug this into water. There's a membrane that's placed on the outside that's permeable to oxygen. It's polypropylene, same stuff that they make saran wrap out of, but these are certain thickness, which you can alter to become either more or less precise. So we have a membrane. Membrane separates water outside from and you can do this in air as well, but we usually are using it in water for aquatic animals. So we can stick the tip of the Clark electrode into the solution, where the fish or crabs or whatever we're working with are, and we swirl them around, because one thing you have to be careful of on the Clark electrode is you can't let it sit static, because it consumes the oxygen. As oxygen passes through the membrane, it is reduced at the cathode, and of course that's what creates the electrons that make the current that we can measure just using the voltmeter. Now, you can make these yourself. If you've got the right chemicals and that sort of thing, they're not difficult. The difficult part, and actually it's not even that difficult, but what you do is you compare it against a meter, and you just use a simple voltmeter, where voltage then is proportional to the oxygen. There's the oxygen concentration in the solution. So we move the tip of the probe around and around so that we renew that boundary layer against the membrane continuously, because if you don't, and you just walk away from it, you'll come back and you'll see that the boundary layer is completely depopulated in oxygen, and your reading is measuring 0. Okay, so that's not what we want to do. So this has been the standard for a long, long time. Again, we used to make our own way back, 60 years ago or so. And then companies started to produce them. The Yellow Springs Instrument Company is probably the most famous, and they have made all sorts of field units where you can take them out. They're really rugged, they're really dependable. As long as you take care of the electrodes and remembrate it occasionally, and calibrate them carefully, then you get some pretty good numbers from it. So I'm going to show you a Clark electrode here in just a moment, and how it's set up. We've got a meter that we can carry out in the field with this, waterproof and digital, and it's got a little computer in it, so it'll calculate a lot of other things in addition to just the oxygen concentration, but typically that's our prime goal. How do we measure oxygen? That's one way we can do it. A couple of other ways that we can do it are something we call redshift technology. I'll show you that meter here in a moment as well. Looks a lot like the Clark electrode meter because YSI tends to kind of converge on a single platform in terms of shape and how the functionality of their meters. But redshift has been around for a really long time. We use that primarily in sewage treatment operations because you have to have a certain amount of oxygenation in the water, right? A certain amount of oxygen in the water in order to know that your treatment system is working properly. Okay, and these would be big sewage treatment outfalls, not just something in your backyard. And so we've been using that for a long, long time, and probably 10 years ago or so, they started putting them on the market commercially for people to use in the field. I've got one that I will show you that I've used exactly once, because at some point along the way, while YSI was developing and investing heavily in the redshift technology, there was another company called Witrox, and they were investing in what we call optode technology. Optodes now have become a little bit more diverse. They are the wave of the future, and pretty much for all of you who become biologists, and if you do become biologists, you will be involved at some point with some metabolic studies with animals. Optode technology is going to be what you will use. The optodes are hugely sensitive. They don't require a whole lot of calibration. They hold their calibration for a long, long time. You can run many, many animals at one time. And again, the computer technology that's been incorporated into that system really helps out. So it Bluetooths all of the information that you collect straight to your computer. So that works out pretty well. And then finally, we're going to spend a little bit of time where we're going to do a Winco titration. Actually, we're going to do two of them. We've got two water samples. One that is came from the we're going to pretend it came from the lake itself, and then one that the fish had been in an area where they would have sucked a lot of oxygen out of it. And so we're going to be able to see the difference anyway in the way that the Winco chemicals work. Now, we refer to the Winco titration as the gold standard for oxygen measures, which means that there is no method more accurate. None of the things that we've talked about, none of the meters that we've talked about approach the level of accuracy that you see in Winco titrations. And if you have a water sample and you are titrating it, you can count the actual number of molecules of oxygen in that sample depending on how much you want to dilute down your titrant. But it's a titration procedure, and most of you have done that probably in chemistry somewhere, but maybe not with trying to measure and dissolve gas in water. So that's what we're going to do. Okay, let's go ahead then, and we'll take a quick break. I'll set up the equipment over here, and we'll talk about it a little bit, and then we'll run our Winco titrations. Okay, so here we have the Clark electrode. This is the YSI, Yellow Springs Instruments. They're found in Ohio. That's where their company is based. And we have been using some form or fashion of this for some time. The meter that I have here is relatively small and handheld. The old ones used to be about the size of a laptop. They had a lot of dials. They were analog instead of digital. But now, because of the miniaturization of circuitry and that sort of thing, there's a little computer built on the inside, so it calculates a lot of different, it has a lot of different functions, and it can calculate a lot of different numbers for you. So let me take that cover off there so we get a better look at it. You can hopefully see some of the numbers that are on there, and there's a lot of things. You can see there's a few buttons on here that we can use. So one of the things that we need to do is we need to calibrate this piece of equipment. I don't expect that you will know or recall all of the steps, but it's quite probable that at some point in the class, or maybe while you're out in the field, you'll have the opportunity to use it. So the first thing we do is we take these two arrow buttons and we can push them simultaneously, and that sets up the calibration feature inside the little computer. Right now, you can't read it, but it's reading altitude. We're in Florida. Our altitude is 0. We are at sea level, so we'll just go ahead and enter that. Okay, and then you'll see that it's switched to a number here. The number on the bottom is 100%. We calibrated this with air. Okay, so we are measuring 100% saturation. Well, that's what our ideal is. Right now it's measuring 91, roughly, percent saturation in air. So it's reading a little bit lower than what we would ordinarily like it to be. So once we've, and by the way, this is in percent right now, saturation, but we can change the mode, and it'll give us, well, it's asking us for salinity there, which is 0 because we're going to measure this in freshwater, so we don't have any salinity. But you can change the mode from to milligrams per liter, right, mg per liter, which is the number that we usually use, and not percent saturation, which isn't as useful. Okay, so salinity is 0. We can enter that, and we've changed the number when we pushed the mode. It changed us to milligrams per liter. So 8.24 milligrams per liter is what we find in fully saturated air, and it bounces around a little bit, but for the most part it's pretty accurate. The second number that you see here is the temperature, and oftentimes then, of course, oxygen concentration in a solution is tied to temperature. The warmer the temperature, the less oxygen you usually have especially in water. Okay, so that's something to keep in mind as well. All right, so we can take this. Here's the probe. This is the probe that we were talking about before. It's got a little guard on it so that you don't damage the end of it. We can take a quick look at how that's put together. And so here's our, you can see the little gold cathode in there, the little wire that runs down there. This is the temperature probe that sits here. Here's our membrane on the outside, and underneath there, although you probably can't see it because it's filled to the top, there's electrolyte solution inside this little area, and the anode sits down. It's that little gray part that you see down there in the middle. So this is a really simple setup. Like I said, you can build your own. They're not difficult to build. They just need a voltmeter, and you've got to have the right kinds of machine parts that you can put together that'll keep the electrolyte and the anode in contact. So that's and then you can just use Saran wrap if you want to for your membrane if you choose to, but you can just buy membranes too online. Okay, I don't think anybody builds their own anymore. It's just not worth it, probably. Okay, so that's what we looked at earlier, and of course that was probably one of the more reliable ways of measuring oxygen, and we use these meters for a good 60 years. And I would argue, well, you can see I've still got one. Actually, I've got 5 of them upstairs that we use from time to time because they make really great field units. When you're done measuring the oxygen, you go ahead and you put this back into the little reservoir on the side, and we've got a wet paper towel in there, and that keeps the membrane saturated and extends the life of the membrane, essentially. Okay, so this one, tried and true, many years, several decades, this dominated in terms of measuring oxygen, and it was a real boon to us because we didn't have the opportunity to measure oxygen in so many different places and so frequently, right? You can measure one measure after another, so it's quick, it's easy, it's accurate, and we still use it. But we've moved on now to better technology in some regard, in that we can do a lot of other types of work and do more of it. And so let's kind of move on to the next thing that we can talk about. I didn't mention, but the YSI meters, usually when we turn them on, we let them sit for at least an hour before we use them. And so they're not as convenient as just flipping a switch, but if you forget to turn it on, you just go sit down and, I don't know, have lunch or whatever, and then come back when it's warmed up. The second type of YSI meter that came out, this really isn't limited just to the YSI company. A lot of people have been using this actually for several decades. And this is what we call the redshift type of meter. Now, it looks a little bit different. We still have a probe that you can see here. And the probe then is tied to the electronics, again, a computer basically on the inside that picks up all the information. This is based on the idea that if you have more or less oxygen in the water, you get a greater or lesser shift in the red wavelengths of light, especially infrared. And so you can measure what we call the red shift. And the amount of red shift you get is directly proportional to the amount of oxygen that's in the water. And so these meters were used extensively in sewage treatment capacity for the most part. But after we began to get a little better at building them, they became accurate enough that we started to use them in academia and research. And so this particular unit was fairly expensive. I would say $700 or $800. We paid for that. I've used it in exactly one study. And that's because some new technology came along and pretty much has usurped it. Now, I will say they still use these extensively in sewage treatment applications and in some types of research applications as well. The advantage of this over the Clark electrode is that you don't have to keep moving the probe. You can just drop it in there and let it sit, and it will continually measure. The other thing, and I probably we talked about it only briefly when we were talking about the Clark electrodes and we were looking at it at the board. As the oxygen moves down and comes in contact with the cathode, the oxygen is consumed in the reaction. And so if you have to be really, really accurate with a Clark electrode, you have to account for the amount of oxygen that's been picked up by the electrode and it doesn't come back into solution. Okay? Now the red shift types of instruments, on the other hand, they don't consume the oxygen. So you get exactly the number that the meter reads. Okay, so they're relatively straightforward and easy to use. The probes are a little bulky and compared to what you saw in the YSI, and miniaturization is, of course, the way to go, because if we can fit more components into the same size equipment, then that helps us out quite a lot. Which brings us then to opto technology, which is, we have two of these units. This is, I mentioned the Witrox company earlier. This is a Witrox 4, it's 4 channels. I've only put the probe on one. So here's the oxygen probe for the YSI. You can see that. Here's the oxygen probe for the blue or the redshift, and this is the oxygen probe for the Witrox. Okay, so it's smaller than not quite a pencil lead, but it's really, really small. It's got an entirely different technology associated with it. It relies on what we call oxygen-sensitive foils. And so there's, you can think of it kind of like a specialized tin foil that interacts with oxygen. They are incredibly accurate. You can, these meters that we talked about earlier, the redshift and the Clark electrodes, they have to be calibrated all the time. As a matter of fact, if you're using them in the field, they recommend that you calibrate them between measurements to keep them nice and tight. You can calibrate this Witrox electrode, our optode, and once you've done that, it's good for probably 3 to 4 weeks before you have to calibrate it again. Now the newest ones, which we don't have, this is third generation technology here, and they recently came out with some improvements. One of the improvements was the new optodes are self-calibrating, so there's no calibration needed. Now the calibration for Witrox optodes is not really that difficult. We put some sodium sulfite into some water, and that strips all the oxygen out. We put the probe in, and then we set the 0. And then we have another usually an Erlenmeyer flask filled with DI water, and we've been bubbling oxygen through, or atmospheric oxygen through that for half hour, 40 minutes, and then we stick it in there, and that gives us the 100%. So we set it at 0 and 100, and after that, good for 4 weeks. So, and in many cases, you can, nice thing about these types of setups is that if you design your respirometer in the proper way, you can put an animal in there and you can measure its oxygen uptake continuously for 4 weeks. It's easy to do that. And people are doing that now. We're looking at really long-term kinds of technology or numbers from that technology. The other thing that we can do with these opto systems is that we can measure what they call micro measurements. In other words, we can take a huge number of samples, and this little box picks up all those samples and Bluetooths it straight to your spreadsheet on your computer. So you can set this to take a measurement once every second if you choose to. The problem with that is you end up with an enormous amount of data that you have to wade through, and I don't find that especially useful. But some people are teasing apart some of the smaller aspects of the overall curves that they get, because animals that, aquatic animals may become more or less active at different times of the day. You may see higher metabolic rates or higher oxygen uptake rates after a meal, things like that. And so it gives you the opportunity to look at segments of the recording and tease those out and decide, or at least identify what's going on in those areas in those times. So that's become fairly popular. I think that in some cases people are really way too far over the top on that, and they'll see a little squiggle in the line, which maybe the fish burped, and now all of a sudden they've written a whole paper about it. Okay, so we try to avoid those kinds of things where we can. But they are hugely useful, very, very accurate, and very, very expensive. So this unit that has 4 channels on it, which you can see, but I'm never going to let you touch, $16,000, and we have 2 of those that we use to run our study. So really expensive equipment, but again, it's really, really accurate, and it gives us a wide range of studies that we can use it for. Okay, so the last type of procedure that I want to talk to you about is the oldest one. I want to say late 1800s this was developed, and these are Winkler titrations, and Winkler developed this particular wet chemistry method for determining oxygen. And so, go ahead and move these out of the way, and we will run a oxygen concentration from 1 and 2 bottles. So here's one that should be more or less fully saturated. This would be before we put the fish or the crab or the shrimp or whatever in there to pull oxygen out. And now we have an after sample that we would have collected from that same animal after the fact. Now we use these quite a lot for metabolism, and not this week, but the following week we're going to have we'll have class review about metabolism and how important it is and that sort of thing, and so we may see this again. But we've got these 2 BOD bottles, we call them, biological oxygen demand. That's what that stands for. It's really, really thick glass. They have this flared top on them, and when we fill them right to the top. And then I'm not going to pull the cork just yet, but there's a glass stopper, and unlike most glass stoppers, they have a flat bottom. These have a pointy bottom, so that when you drop them in, and you always drop them in, you don't place them in. You set it up there, drop it in, all the air bubbles get pushed out. And if you get an air bubble in there, depending on your need and your experiment and the precision associated with your experiment, you may have to do the sample again if you get an air bubble trapped in there. So these bottles then help to prevent that. So these BOD bottles, quite expensive as well. Usually they sell them for about $300 for a dozen of them. Okay, so again, we try to be careful with them, but you can see from that one right there that some students are less capable of handling glass than others, I guess. Okay, so based on what we've got here then, let's go ahead and we will do our first liquid titration. So let's do the saturated water first. Okay, so when you are using, and you can see these are all numbered because people use them for light-dark studies too. You drop them on the bottom of the lake and you fill them with lake water and you figure out oxygen demand and that sort of thing, or seawater demand. Okay, so when you pick these up, you don't want to contaminate anything. You usually grab the cork between your fingers and pick it up so that you're not getting anything, because you don't know what's in here, or at least you might not know if you're a technician, for example. So the first thing we're going to do then is add the manganese sulfate, and we need 1 ml of that. You can use and this just has to be approximate, doesn't have to be exact, so we'll do that. We stick that into the bottle and shoot that in, and try not to get an air bubble in that. There we go. Okay, and then, like I said, we just don't want to do that yet. One more. Second one. Second thing we put in here then is the potassium iodide solution. Again, 1 ml. All at the bottom. Put that in. You can see that the reaction is picking up the oxygen and turning it into, oops, I didn't mean to leave that in there. Okay. And now we drop that back in. Empty the excess off. And we roll the bottle, we never shake the bottle. Okay, what do you see in there? The orange flocculant, we refer to that as flocculant because it looks like a cloud in there. That is the oxygen bound up right now from the potassium iodide and manganese sulfate reaction. Okay, so we roll that bottle around, okay, like so. The bottles are built so that they roll the flocculant around to try and pick up as much of the oxygen as we can. Usually do that for maybe 30, 40 seconds. Okay. You never shake it because it takes all of, now sometimes you have to if all the flocculant doesn't get used up in the reaction. We have to shake the crap out of it to get rid of the little pieces that are left. But in general, we let that settle down to the bottom. That's picking up all the oxygen that's in that system. Okay, so now we will do the second one the same way. Bottom of the, or close to the bottom anyway, we let that go. Here. And there's no need to, we just got to make sure that we've got enough of the solution there. There's no need to measure it out really, really precisely. We put in more than we really need. Okay, this is our fully saturated bottle. Here's our bottle that contains far less oxygen, and you can definitely see the color difference in those two. In fact, I'll be surprised if there's any oxygen, free oxygen in this bottle. But again, if this were a metabolic study, this would probably be the water coming out from the respirometer where the animal is consuming oxygen. And this would be maybe the before. You can take the before and subtract it from that, or the after, and you can end up with measure the total oxygen uptake over your time period that you would have set that out. Okay, so we'll let that sit for just a moment. You can see this one's pretty well settled out. The flocculant's kind of all gathered up at the bottom. And once it reaches that point, you do it one more time. You roll it around to get that last little bit of oxygen in there. You want to get like all of it. Remember, if you do this correctly, you can measure every single molecule of oxygen in that bottle. Give that one a little bit more time, but you can see it settling out too. Okay, so now what we need to do is titrate these samples. You can see that the flocculant is all settled out, right, in the high concentration, oxygen concentration bottle, and you can see the flocculant looks quite different, but still, there it is in the low concentration bottle, okay? So, what we're going to do now is we're going to add concentrated sulfuric acid right there, and it doesn't matter how much of that you add, but this is going to fix the sample. Once the sample is fixed, no more oxygen can get into it, and so that's the key here is that we're going to fix that sample, and you can set it in the refrigerator for a month or so if you don't have time to titrate it at the time that you collect it. We usually titrate them soon after as soon as we're done with the flocculant stage. Okay, and again, you don't have to really measure out too much about how that works. There is a trick though when you take the cap off in this case. You want to make sure that you run the acid along the ground glass surface, and watch what happens to the flocculant when we do that. There we go, and again, flop that back in. Dump off the excess. And now, we will have one more reaction. We do have, we'll keep rolling that, and you can see that we've got some fuzzy stuff in there, so it could be that we'll have to shake it here in a little bit, but if we keep rolling it around, we will eventually turn that into a kind of a clear amber color. And this is what we're going to titrate. Okay. Impressive so far? All right, we'll do the same thing again. With the low concentration. And all the way around the neck. And drop the lid back in. Pour off the excess. And it's probably going to come out with 0 oxygen in it. But we're going to titrate it anyway. We'll see. Maybe there'll be a few molecules left in there. Okay, we can shake that. As much of that as we can. All right. Now, let's do this. Close that up. Spill that. Okay, so the BOD bottles hold exactly 300 milliliters. We are going to titrate a 200 milliliter sample of this using sodium thiosulfate. And the difference between where we start and where we end up then is directly proportional to the milligrams of oxygen. Okay, so we can read it right off the burette when we're done. We always do 200 and not the entire bottle, because sometimes you've all done titrations probably before, and the first time you do them, you run right past the end point, right? And in many cases, this may be a really valuable sample. Maybe we've collected that for a whole week and we can't afford to lose that. So we do 200 first, and if we screw that up, there's still 100 left, and we just double whatever the titrant value was then. So let's go ahead and we'll 200. Milliliters. Okay, so here's our sample, 200 mils. This is our titrant. We're starting at 13 on the burette, and we're going to lower that down until we clear this sample. When it goes clear, then we know we're done. And the difference then between where we started and where we ended is directly proportional to the oxygen concentration in milligrams per liter, right? So our milliliters, we've set the chemistry up that way so that it's easy, right? You just take that subtraction, that's your oxygen content. Okay, and so initially, you can turn these things up pretty quick, and there we go. You can slow it down a little if you want to, but we're just gonna go ahead and add titrant to that. And you can see it's starting to get a little bit lighter as we go along. You don't want to overrun it, so once it gets quite pale, let's slow that down a little bit. There we go. Okay, so here's what it did look like, and we're titrating it, and it's getting lighter in color. So now we'll just do it a few drops at a time. A bit more. I'd like it to be a pale yellow. I'd like it a little more pale than that. Everybody's got their own preference, I guess. I do a lot of these, so it doesn't bother me to go a little bit further than other people might. Okay, you can see that's pretty pale there, and what's going to happen is it's going to get to the point where it's going to be really difficult for us to see the transition from this really pale to completely clear. And so what we can do is we'll add a second indicator. And this is starch. And now we'll titrate that to clear. And typically it's a lot easier if you haven't done these before, but where you can put a white piece of paper behind it to be sure that you got the last little bit. But for us, it shouldn't be too bad. This is always the part where people run over because you get anxious and then you think that you should be moving faster, but it's getting there.
It's a little harder to see on the video, but it is getting lighter as we're going.
Mm-hmm. Should put the towel on there, that would help too. a little bit. Should be coming pretty quick. You can see there's a few little particles left in there that I hadn't counted on, and that might be a bit trickier for us to. It's pretty close. See, that's still got just a little bit of pale purple in it. Now we're down to that point where we add a drop or two, and that should get us where we want to be. Let me say that's pretty close right there. Okay, so we started with 13, we ended up with 20.2, 7.2 is our difference, is that right? I think so. And when we were measuring oxygen saturation on the Y side, it was 7.4. So good corroboration between those numbers. So this is, again, this is the standard for measuring oxygen. Typically, if you do this as in a study, you get really fast at it because you kind of know where the numbers are anyway, and so you run it right down to very near where the bottom is, and then you start doing the final part of the titration. So these methods then for measuring oxygen have been hugely important in understanding biology for a whole lot of reasons, because we're constantly measuring oxygen, as we noted, in things like streams and lakes and body fluids from different animals and things like that. So it's a really useful technique to be able to understand the options that are open to you based on A, how much money you have, and B, how much time you have. But a good working knowledge of instrumentation and being able to utilize oxygen measurement techniques is essential to metabolic studies, and metabolic studies are the heart and soul of biology. If you are a biologist in any way, shape, or form, somewhere along the line, metabolism is going to be important to you, because that's life itself right there. How we use energy, how much we take in, what do we do with it after we've got it, how does it influence our behavior, our adaptations, everything comes down to that. And in order to measure that, you need to be able to understand how oxygen is measured. So with that, we'll go ahead and close down shop. Tyler will send out a worksheet maybe and ask you to do basic review of concepts that we covered and send them back to him. And I think we're good for this week.